So. All right. All right.
All right.
What's first on our list?
All right, let's get going.
Get going.
Niskin bottle.
Niskin bottle.
Okay.
Yes.
Ooh.
Okay. A few meters off bottom.
Should they go to sample or?
We can do, just switch over the camera to, oh no, it's already there. We should be good. We can, when I get the arm out, you can pan the camera over to the Niskins. All right, go ahead.
All right.
Pan left. Oh, it might be slow.
Turn it up.
Yeah, turn up to give it some more beans.
More beans.
So slow.
It's very
Oh,
slow.
we can adjust that
Yeah.
in a bit. Arm is very shaky.
Right.
Yeah. Well, sometimes, like, if it gets into a certain position, it shakes.
Yeah, it also has. Oh, I'll go for it. Going for Niskin 1.
Yep. Yep, it went.
It did?
Yep.
Oh, all right.
Which number is this, May?
This is number 16.
16.
Panning back right.
Yep. Can you note down the red book that after that the craft is shaky?
Mm-hmm. There. Tilt down.
Yeah, sure. And, you can
right.
tilt up to basically, or pan right so that it's square, and then tilt up just so you don't see the porch. Yeah.
Okay.
Nice. All right.
All right, you ready to get going now?
Ready to go.
All right. We're going to go 160.
160. Sounds good.
Bridge nav. Good afternoon. We are ready for a ship move. We'd like to go 20 meters, 160 at 0.3 knots.
Would you like me to leave the valve?
You can lower it down to maybe 1500.
Thank you.
seems to be the sweet spot for that valve pack.
So a viewer was wondering what probe was just taken by squeezing or moving those colored balls.
Those are water samples.
So that triggered one of the Niskin bottles. It collects water samples for our eDNA stuff.
Goes here.
Yeah, I might try cycling power to it, but oh, that was the wrong one. Text blank.
Yeah, just to orient our viewers a little bit too, so we acquired bottom here, 2,300 meters, and we're going to be diving off the flank of this ridge. The area, the landing spot we chose is a bit more gradual slope here. You can see it's heavily sedimented, a lot of sediment here. The first part of the transect is going to go over this more gradual sloping terrain, but then it's going to start inclining quite a bit more. So about 200 meters or so from here is where the steepest part of the ascent will start, and that's where we expect the sediment to be much, much less so.
Zoom on this little shrimp. Oh,
Oh, it said no.
said no thanks. No zoom.
It's a shy shrimp. This is a Aristeid. See that long rostrum?
All right, come on.
There might be something on the rocks.
Check it out.
What do we got here? Little black coral.
Yeah, it looks like stauropathes. 2 colonies I see.
There's a few of them.
Yeah, there's 3 of them. There's another coral on the other side of the rock too.
Yeah, so black corals there, a group about 250 or so currently described species, and many, 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 many more to be described. They are the closest living relatives are the scleractinian or hard corals. Now black corals, the vast majority of them are adapted to live on hard substrate, so they have a hold fast and when grow on rocks like this. There are a couple groups that live in sediment. They have a modified hook-like hold fast that allows them to grow in sedimented areas, but in general they're all found on hard substrates. See some really nice sand ripples there.
There's a sponge.
Yep.
What's the purple?
Oh, it's a Paleopatides, a type of sea cucumber.
Oh, cool.
This is an interesting sponge.
Where's the look?
Yeah, I think it's worth a look.
Okay.
It looks like it has a
Push in a
little
bit there.
feather star on it as well.
Tilt down on the camera a bit. Sarah.
So just like the Antipotherians you were talking about that root in the sediment, this sponge is rooting in the sediment. So it has these long, specialized spicules, these pieces of glass that it uses to root itself down into the sediment.
Oh.
This is probably a faranomatid glass sponge. Probably Hylanema. Nice.
Okay, come wide.
Is the black dust around the rocks manganese?
It could be, yeah. So little pebbles.
Yeah, most of the boulders and rocks that we see around here probably rolled down a long time ago, kind of crumbling.
Yeah, if we can get a quick view on that fan to our left there.
Oh, that one. Okay.
Yeah, I think that one is Romilogorgia militaris, a type of chrysogorgid coral. It's pretty common at these depths. They usually live deeper, which is why we didn't see them yesterday.
I presume there.
Bridge nav. Yeah, they're really pretty, these corals. They have very delicate looking polyps. The way to tell them from other chrysogorgids is that the branches all line up in a line, and that's how I remember its name, militaris.
Yeah, that's great. Thanks, pilot.
All right.
Yeah, so we'll likely see a lot more of those along this transect. They're quite common.
Yeah, you can see some really nice sand ripples there, and we're pointed here almost due south. And so this gives you a little indication of the predominant water movement. So going here from left to right, so between east and west.
We can check out that sea cucumber real quick. This is a sea cucumber in the family Synallactidae. Also a very common animal to see at these depths. They're kind of like the vacuum cleaners of the deep sea. They just meander around and eat sediment and consume the organic matter
Go for a
and
quick zoom there.
poop out really nice clean sediment. That one's a pretty one.
Are the pink things like a part of its intestines?
Wait, which things? What did you say?
The pink thing, like inside.
Oh, that like little pink area?
Yeah.
Could be its stomach.
Um,
Oh, there's a little black coral there. You can see its intestines too through there where the sediment filled. Yeah.
Excellent. Yeah, that's great.
Shall I zoom out?
Yeah,
Yep.
we're good here.
Yep, all set.
I'm just messaging Mike about the cinema cam. Not going showing up. Or maybe he can hear me if he's in the data lab in the hole.
Yeah, it didn't come up when you cycled it, did it?
No.
Mm-mm.
We've got one of them, don't we? It's the other one. Yeah.
Okay, moving on.
Yeah, let's try to cover some ground here.
Yeah, should we do a larger jump this time?
Yeah, I think
All right.
so.
Bridge nav. Can we move 40 meters 160? Thank you. See, there's more of that Romilogorgia. There's a little Calificus. And some more Stauropathes. And there's a very small Chrysogorgia, very sparse.
Nice.
And a small shrimp. I wonder if we'll see some fun sea pens in the sediment.
And so to all our new listeners and viewers, we are approximately 70 nautical miles north of Alaska Reef and have just reached the bottom of the seafloor. We are at a depth of approximately 2,274 meters.
Yeah, thanks, Imade, and we're going to be diving here for the next 10.5 hours or so, climbing. So the habitat we're expecting. So it's a initially gradual terrain here for the next 100 meters or so, and then we're going to go into a lot more steep terrain.
Oh, there's a sea pen.
Yeah.
Sea pen. For a zoom?
Antiptylan?
No, I don't think
Question
so.
mark? No.
Let's see, it's pinnate. Do we have more zoom?
We do, but it's going to go out of the frame. There we go.
Huh. This looks like a black coral. Not a sea pen.
Yeah, I think you're right. It's just we're running from the side. I'm very puzzled there, yeah. But I think...
Like a heteropathes.
Yeah.
Oh, it tricked me, see? I was like, oh,
Yeah.
it's a sea pen, but no, it's not.
The sediment, I thought they attached to like hard substrate.
Well, I think there's actually some rocky underneath it, but I think there's actually might be a collection candidate. If we're,
Okay, I'll stop the ship.
I'll zoom out.
Bridge nav. Can we hold position? Thank you. See, that's why you put in the big moves, so we find something really cool. Because if you do the short moves, then you won't find something cool. Because you're playing it safe.
Yeah.
Take chances,
You have to
get
commit.
messy.
Yeah.
What are we thinking for this sample? Where do we want to put it?
So these things tend to be
Floaty?
floaty.
So is it maybe a snip and slurp?
I think you'll end up getting the whole thing.
Okay.
Because they're very wiry and they don't like to snip.
Okay.
Yeah, so I think if you just grab it and slurp it, if it comes up with a rock, we can stick it in a box. If it doesn't come up with a rock, then we'll slurp it.
Okay.
If we do put it in a box, I think...
Yeah, we got it. Dan, thanks.
If we put it in a box, Jake, I think it would be best to put it in one of the little ones in the back of the starboard bow box, just in case it's floaty.
Okay.
Okay.
Yeah. Gabby doesn't like these.
That's not.
But they do slurp nicely if they don't come up with rocks.
Do you want some zoom?
A little bit of zoom, yep, push it.
Oh, it's snipped.
Snipped.
Huh.
So maybe that's a good slurp?
Now let's slurp it. Otherwise it will
Yep.
fly away.
Okay, come wide a bit, Aaron. Can you pan the camera right
Yep.
towards the section? And then down a bit. Nice. And then...
Turn the pump on.
Yeah, turn it on.
Okay.
I don't think we're close enough, but... Oh, we'll get some
Oh.
sediment.
Oh.
Here, it might be easier if we just
Might
pop up.
pick up. Yeah.
All right, stopping. So we don't get too much in there.
Oh, we don't have the chamber camera up.
Oh, yeah. Let's go to
Sample.
sample. And then switch over one of the starboard cameras. Yep, I think the starboard camera to the bucket.
Got
There
it.
we go. Now we can switch to chamber 1.
All right. Let's go. Okay, rotating. Let's go the other way to number 1. There we go. All right.
Cool. Oh. Is the suction on?
Suction's on, right?
Not yet.
You can turn it on.
We had shut it off since the sand was getting pulled in. There we go.
There it goes.
Yay.
Very nicely done.
Got it.
There
There
it
it
is.
is. All right, stopping.
Let's see if that one is the same thing.
Right there.
Just out of curiosity. I think it is.
Go for a zoom.
Yep, same thing.
Yeah,
Cool.
that's
Interesting.
great.
All right. Go back wide.
Yeah, but I think they're actually on hard substrate that's just lightly buried.
Yeah. They're just growing on small pebbles or something.
Yeah. But yeah, if that genus is alright, yeah, that's a genus that has not yet been recorded here from Palau.
It looks a bit different from some of the other ones that I've seen too.
Yeah.
There we go.
Yeah, but that gives us an indication that there's probably a pretty light sediment cover here.
Yeah.
Stocked crino.
And that's a good,
I think it's on something.
yeah.
Yeah. It's very pretty. Oh, it's on Iridogorgia. You couldn't even see it. It blended into the sediment. Wild. This is a Sathermetra. Saw some of these yesterday. And then got your Iridogorgia. Bridge nav. Yeah, we'd like to move 30 meters, 160. Thank you.
Yeah, it looks like from the high pack about 100 meters, or well, a little less than 100 meters before it starts inclining a bit more.
Yeah. Kind of just want to get us to the base of those hard rocks.
Yeah, agreed.
I have a feeling with how much we've seen so far that it's just going to get way better, like yesterday's dive.
So I know the sediment core idea was like a maybe, and there was this interest about it being near kind of gravelly manganese. I don't know if it's worth doing that, but
I think this sediment's too coarse and it's not going to stay in the tube.
I agree. I'm just thinking aloud because this was a request.
Yeah.
But
Yeah, I would concur, like, yeah, that unless you scoop it, yeah, you're not going to be able to get it. So yeah, maybe that's something we hold off.
Yep.
Oh, let's look at that anemone.
Sure. And a shrimp.
Oh, yeah. I think it's an Actinurnus anemone.
Go for a zoom.
I like its puckered mouth. Very pretty. We're going to get so many good shots for animal guides and stuff. You guys have been rocking it with the zooms. Can we pan down to the shrimp?
Sure. Sarah, you want to tilt down on the camera? Up one more. There we go.
Oh, there it is. Oh, there's a little sponge too.
What's that little?
Like a shrimp.
Cinematic the long-legged shrimp. A small glass sponge and a small chrysogorgia.
That's great.
Cool.
Very nice.
All right. Come on.
Do you think we could get some lasers on?
Oh, yeah. Sure.
Yeah.
One of our scientists to shore is Aaron Judah is requesting it as well.
Oh, did Aaron get into the chat?
Indeed.
Awesome. Hi, Aaron. Aaron works in the same office as me. We share an office.
Aaron says hi back again, and hi Ika, thanks for joining us. She also says hello to everybody on board.
Oh, there's an umbellula. Do you see it?
Right there.
Yep.
Come down a little bit. Stretch the tether out a little bit.
Nice.
So the two lasers are to get the approximate size, is that correct?
Yes, the lasers are 10 centimeters or 4 inches apart.
Go for a zoom there.
So for example, this umbellula is probably about 15 centimeters long. Umbellulas are a type of sea pen, and they have a really fun name.
Can we tilt down a little? Oh, there we go.
Did a little slide.
Do they ever grow larger than 15 centimeters?
Oh, absolutely. In the little video clips that we were playing before we started getting underway, there is Umbellula monocephalus that's like well over 1 meter long.
Oh, wow.
Yeah.
Yeah, that's a great shot. Thanks.
Okay.
Analysts on the move.
Is that last ship move complete?
Ship move is complete.
Okay.
Just thinking about another one.
Yeah.
Yeah.
Send it in.
Bridge, nav. We would like to move 30 meters 160. Thanks, you're the best. want to see the wavy stick.
Where?
That, yeah, right up there. Yeah. Is it a dead sponge with zoanthids on it or something else?
Go for zoom.
It is definitely zoanthids. So my best guess is they're growing over the stalk of a dead sponge, kind of like the one we saw earlier.
Excellent, yeah, we got a good shot of that.
Oh, and there's a small bryozoan on the rock too.
I just see sand. I don't know how
Did
you're
you
looking
see
at.
sand? You just have to believe me. There's
I believe you.
there's a brittle star underneath the rock. You see its little arms?
Oh yeah, I see that. All right.
Brittle stars are really common. The more you look, the more you see. They're really good at just being everywhere.
Yeah, particularly under rocks, if you were to flip any of these over.
Oh yeah. Just like when you're a kid and you just flip rocks for bugs, the brittle stars are the roly-polies, you know. You find them everywhere under rocks. There's something floating in the water. You see it, the little black thing?
Black thing.
It's upper right.
Oh yeah, what is that?
I don't know. Jelly? I think it's a jelly.
Go for zoom.
Oh yeah. Small little jelly. It's a pretty one. Kind of looks like a flower.
It's so small, so it makes it look like it's going so fast.
Yeah. Well, to it, it's probably moving super fast.
Yeah.
Nice. All right.
That's really cool. Yeah, those lasers. It's like 2 centimeters.
Oh, there's something. Oh, shrimp?
Jeff is interested in slurping that up.
Okay.
All right.
What do you think about that, Daniel?
Yeah, absolutely.
Our ship move is complete.
But it'll just stop right there.
Brian agrees.
Might be able to, if you turn on suction here.
Okay.
Might be able to just
Turning on.
process myself.
Are you just going to fly it in?
See.
All right, suction's on. Oh, have to see what sample jar we have, though.
If I can get low enough.
Should I bring up the sample? Hello?
Yeah.
I don't think I can get low enough at this angle.
Sorry, I don't want to mess with you.
Oh. All right.
All right, bucket up. And which number would you like?
2, please.
Okay. Wrong way. There we go. Nope. Hates me.
All right, just suction on.
Yep.
Suction's on.
Oh. Buried it. Buried it.
Oh, there
Got
it
it.
goes. Oh, it's in there. Turn off. Otherwise, you're going to turn it into mush.
Let the dust settle. See what happened.
Can you rotate it back to the flush jar?
Okay. Back.
Yeah, then you can flush the sediment that might still be in the
The hose.
hose.
All right. Turn on suction.
Yeah, there's a lot of sediment in there.
Yeah.
Yeah, look at all the exhaust.
All right, that should be good. Come up a little. Want me to bring up the, is it dive or ROV
Sure.
cams?
Yeah, the blank one. There
Okay.
we go.
Perfect.
Which jar was that in?
2.
Listen to, I can pan back over.
Yeah.
Hold up. See if it has settled down now.
Not yet.
Yeah, it might take a while.
Can let it rest there if we want.
And it's best not, because the thing could find its way out.
Yeah, there's a
Oh, yeah.
opening. So
Okay.
go back to flush.
Let's go back. Just stay on flush.
ridge, Nav. Can we move 30 meters 160? Thank you.
160.
Oh, it's a small polyopogon. Look how cute it is. Those sponges can get actually really huge sometimes.
Really?
They're just like the really like meter and a half wide.
At the end of my leash.
Yeah.
Tugging.
I can come down a tiny bit, but
That's alright.
okay.
Stay there.
There's something
There's
floating.
something in the water, sea cucumber. There it is. I see the shadow.
Oh, it's up. There it is.
Probably Hypniastes or something.
Go for a zoom.
Oh,
That is
it's
awesome.
like a Amphoraema or a Peniagony. It's in the family Elpidiidae, a type of sea cucumber that often swims.
Yeah, that's great. We got some really nice stills there.
Yeah, so they'll land on the bottom and eat, and then they'll jump up and float somewhere new and land on the bottom and eat.
What do they eat?
They eat organic matter that's in the sediment. So they basically eat sediment.
Which is a very low nutritional value, so they end up eating a lot of it.
Can I go back in?
Yeah, you'll notice these little trails on the seafloor. Those are often made by sea cucumbers.
You happy for me to just zoom again?
You can go back in.
There's a different type of sea pen. I love how they swim. Just sort of slow crunches. This one's so see-through you can see its gut.
So graceful.
Mm-hmm. can we take a look at the
Yep.
sea pen on the seafloor?
Come on.
Yeah, that
That
one's different than the one we
right.
were looking at before. I believe this one is called Copoblemnon.
Go for a zoom.
Yeah, that's great.
You can see the pinnules on the tentacles of the polyps. That's how you know it's an octocoral. They have 8 tentacles, and then they're always pinnate, little side branches. Every octocoral has polyps that look like that.
All right, so I think we're about to get into the more steeper part of the terrain here.
Okay.
So can keep us moving along. So I'm really hoping that by the end of this watch we can get past that waypoint 2, if possible.
Well, I'll try our best.
Okay.
All right, gauges, pressures, all good.
Nice.
Oh, what's that? It's fish.
Fish. Little fish.
Looks like a midwater fish. But maybe not. It's wiggly. What kind of fish are you?
Go for a zoom.
Oh, it's a super tiny rat tail, I think. Or not. Maybe it's ophidiid.
Ugh.
Do you see his dorsal fin?
Not yet, no.
Looks a little bit like the fish we caught.
This one looks different than that, to me.
Maybe it's just its size. It's hard to see at this
Yeah.
angle. Oh, there. Just
Okay,
turned.
there we go.
Nice.
Yeah.
Oh,
Yeah, this-
it kind of looks like Porogatus.
It's got a big mouth.
A type of ophidiid. It is really tiny.
Yeah.
Yeah, great eye.
Couple centimeters.
Oh, there's big rock. There's something on the big rock.
Large sponge.
It's a dead sponge. But there looks like there's something living on it. Sometimes
Tiny
there
hydroids.
are cereanthids that live inside these dead sponges. The Batra nucifer.
Go for a zoom.
Oh, yeah, there's some serpulid worms, polychaete worms. And then there are cereanthids. The cereanthids are the purpley ones, and the polychaetes are the feathery, tubey ones.
Cool.
That's great.
All right,
Yeah,
come on.
cool.
I think I saw another coral right here.
Bathypathes. So this is a black coral called Bathypathes.
Yeah, these are always, they have a series of polynoid worms that live pretty much always in that, if you were to zoom in on that center channel, there's usually some that are nestled amongst those.
Go for a zoom.
This looks very similar to a species that we have in Hawaii.
Mm-hmm. Bridge now.
So this group of black corals, they have
Can we move 30 meters?
very
160.
elongate polyps. A family of that has the unfortunate name of the Schizopathidae. Yeah, so it's hard to see because you can't really see the mouth now, but they're elongate, and every polyp would have 6 tentacles. But in this family of black corals, they're so elongate that it almost looks like they're polyps with 2 tentacles because they're very stretched along. And yeah, this is one of the more common genera in the deep sea.
Okay, come on.
I always like good Bathypathes.
Yeah, quite a contrast to yesterday's dive. And this is all part of trying to explore a wide range of depths. So tomorrow we have an even deeper dive planned, approaching the depth limits of Hercules, so going close to 4,000 meters.
Oh, is there?
To the left, up there.
Oh yeah, good,
Yeah.
good eye. Let's chase it.
With the net?
I don't know.
Not yet.
I'd like to know what it is first.
Yeah, I know Jeff's watching.
a cusk eel. Looks like Spectrunculus.
He wants nothing to do with us.
Yeah, that's fair. We don't have any snacks. If we had snacks, I bet it would be more interested.
Go for zoom.
Jeff says it's a Lucio brotula
Oh, is it?
because it has a scalloped forehead, so it can't be a
Oh.
Spectrunculus. Okay. Jeff would know. I always have to ask Jeff about these.
Yeah, thanks for those IDs.
All right.
Got like a little coral or something over on the rock to the left. I want to say a sea pen maybe, but
Since it's on a rock, it's unlikely to be a sea pen.
okay.
But there are some sea pens that do root on rocks, but they have these big inflated bases that act like suction cups,
Oh,
and they tend to be red.
okay.
This might be a bamboo coral. That's my guess. Once I see the polyps, we'll know for sure.
Go for a zoom.
Yep, definitely a bamboo. You can see those black bands on the skeleton through the tissue. The bamboos usually have these little red mouths and large polyps. Very distinctive. So this one's either a very young unbranched one or one that has not yet started branching.
All right. Come on.
Excellent.
The other long wavy thing in the water there. Probably another bamboo. Now that I see it better. Or maybe it's a Candidella. Looks very skinny.
Go for a zoom.
Ah, yes, this is Candidella gigantea, a primnoid coral. You can tell the difference if you're looking at the polyps. You see how these polyps look a little bit scaly. They're not as big and fleshy, and they don't have the red mouth like the bamboo we were just looking at. That's how you tell a difference. So Candidella gigantea has whorls of generally 3 to 4 polyps. They grow very long and have relatively large polyps for a primnoid.
All right.
He changed heading.
Okay.
I saw him do it and didn't think it would be a problem, so.
Coming up a little bit.
Roger.
Yeah, Megan, do you have a sense of the range to waypoint 2? So like 160?
Yeah, so it looked like 300 meters.
Oh, wow, okay.
Yeah. It's a little bit further than it looks on the map, huh?
Yeah, quite a bit. So yeah, maybe we can cover a little bit if we can.
Yep. I think we've been making some pretty good progress. So far,
What's halfway there?
we've gone about 275 meters.
So a question for the science team. What would you say is the most common plant or animals in the deep?
Well, there aren't any plants down here, so none of those. Well, the most abundant animal in the deep ocean is the bristlemouth fish called Cycloxone. It's one of the most abundant vertebrate on the planet. We often see them during dives, but they're considered a midwater animal. We usually see them in the water column. But things that we see quite often are highly variable depending on where we are and what we're surveying. Some areas are very densely populated with corals, where others are densely populated with sponges. Echinoderms tend to be the most abundant taxa in areas with sediment. Or just like in general echinoderms, like our sea stars, our sea cucumbers, sea urchins, sea lilies, all those types of animals are generally the most frequently observed.
Are they done with their heading change?
Yeah, he's done with that. I'm going to get us moving again.
Cool.
Yeah, it really did.
Bridge, Nav. Can we move 30 meters, 160? Thanks.
Yeah, and it really depends on the scale we're looking at. Obviously, this is we're mostly focused on the macro fauna, on the things that are a little bigger in terms of numerical abundance. Obviously, it's the really tiny things that you would need microscopes that would be by far the most abundant. So yeah, in these sediments, they're always worms, all kinds of polynoid worms and polychaetes and bacteria would be in tremendous more abundance, but yeah, for the bigger things. And that's also one of the...
Can we snap on that coral that's on the rock? I think that's the same one that we collected. Yeah, yeah.
Good eye. Yeah.
it looked like it could be different. I thought it was like branched, but it was just a shadow.
Have resume.
Yeah, just a quick look, and that's good. We saw that one before, and we collected it.
That's great.
That's the big plus about being out here and getting to ask for these Zoom requests is when I go and annotate the dives later, I won't be like, oh, why didn't they look at that? Because if we don't look at it, it was totally my fault. But it helps answer some of the questions that we have. We can get more accurate identifications if we look at things at least once. Oh, there's a crinoid? Pentametricrinus. So it has only 5 arms.
Have resume.
Nice.
Kind of
Maybe.
5 arms.
What does it mean to annotate a dive?
So we use a program called VARS, and it helps us log the occurrence of every animal that we're observing. And in addition to just the animal name, we record associated data, including information about the type of substrate that we're observing in the area, what the animal was observed associating with. So it could be another animal or a rock, a cobble, bedrock, what substrate was it seen upon, as well as information about interesting characters maybe or behaviors that it might be exhibiting. So we take a lot of these notes and that creates a small little database just for this dive that can be used by scientists to enhance their understanding of the density and diversity of animals in a certain area or across an ocean basin. So you can look at the data across many, many dives or just a single dive, maybe one taxonomic group to better understand distribution. So we create this data product to help all of us understand these deep sea communities better. And you'll be able to ask questions and use the data to answer those questions. As is, when we watch the dives, they're very, very beautiful, right? But before we annotate, it's not analyzable in a meaningful way. You can say, oh, well, I saw this, but that's a little more anecdotal. Annotating puts the visual data into more of a numerical format that we can run statistics on.
Keep on moving.
Yeah, I guess so. Bridge nav. Can we do another 30-meter move, 160? Let's look at that one. I think it might be a cophenomenon.
A what?
Another sea pen. Yep. Yeah, we saw one of these before.
Yep. For a quick zoom on there. The darker part is like this, a skeleton or a limb or
that's the gut.
the gut.
Mm-hmm.
Huh.
It looks like there's a barnacle on it too.
Oh, yeah.
Good old gooseneck.
Yeah, the skeleton, there's like a little bit of a pen straight through the up and down axis. But yeah, that dark purple is the gut. A lot of animals will have like a red or dark colored gut, because if they happen to ingest things that bioluminesce, you don't want to be seen.
All right, come wide.
A follow-up on the annotations, are those available to the public?
Yes, absolutely. All of the data that we produce at the Deep Sea Animal Research Center is available online for download and use. We are contracted by NOAA OER, the Department of Ocean Exploration and Research, to annotate these dives. And they're the ones that take the data products from us and make them available on repositories like OBIS and the NOAA Deep Sea Corals database.
Yeah, looks like we're just about at the base of this more sloped terrain.
It's still a lot more sedimenty than I expected. Could also be where we are on this feature, kind of to the northwest, depending on the currents around here. Maybe there was like a little bit of a slump or something. I bet by the time we get up to waypoint 4, it'll be different.
Nice large Bathypathes colony. Yeah, this is good sign here.
We're getting more rocks. Yeah.
Steeper slope. That sonar.
Speaking of rocks, are we getting any rock samples this time?
Yeah, we should probably grab a rock sample before we get too shallow.
Yeah, just
Um,
all of this looks like pretty much tumbled down.
Yeah, we want to collect something that is close to, solid bedrock.
I haven't really seen anything that looks like a good candidate so far, to be
Yeah,
honest.
Yeah, I agree.
me neither.
I think everything just looks like it's crumbled. Not really in situ.
Probably soon. I think we're going to get close to something that might be better.
And then maybe it'll be carbonates, but
Hopefully not.
we'll see.
We're still deep enough that there shouldn't be any carbonates.
Let's see how crumbly it is for how long.
But this piece here looks
Yeah, that
not
looks,
like that looks more in place.
definitely. That's starting to look a bit more in place, yeah. Very cemented.
Do we know what kind of rocks these are?
These are basalt.
Basalt rocks.
And there's manganese crusting over the outside. You can see that the sort of round kind of dots, that's the manganese crust forming its botryoidal texture. Bridge nav. Yeah, can we move 30 meters, 160? Oh, there's another one of those Romilogorgia militaris.
Over zoom.
Looks like there's an anemone on the couple anemones on it. And there's a brittle star. Oh, and there's ice pod.
Nice. Okay. Come on.
That's great.
Pathy.
Yep, that's right.
Jake's on his way to becoming a biologist.
One at a time.
It's kind of my goal to make everybody a biologist by the end of the cruise.
for those that are following along with the dive plan for our scientists ashore, that we send out the dive plan. So it looks like we're somewhat in the midpoint between waypoint 1 and waypoint 2. Current depth of 2186 meters. So we'd set up a pretty ambitious track length in the dive plan, close to 2 kilometers. We probably won't reach that, but we'll try to cover as much ground as possible. We do have another 9.5 hours of bottom time. We'll be, of dive time, so we'll be up on deck at midnight.
So something like a little, if it were possible to break off like a little edge of one of these pieces of rocks, or something that looked like it was already cracked, that might be a good candidate for a rock sample.
Okay.
I'm thinking more just sort of out of view in the right.
Right up here.
I think those will be too hard, but it's now a little bit out of view, sort of behind us. Yeah. Yeah, like those little ledges there. Yeah.
That really square edge.
Yeah. Something where you could actually grab it with the manipulator. Something, yeah, like these little pieces, exactly. Those might be really hard to break off. It might not work, but they might crack off, and that would be a good candidate.
Oh, and there's the Stichopathes.
Yeah, or that little lip that's sticking out on the, like, right around there, yeah, and there, or to the left of that as well.
Okay. Give them
Kind of any of those little
Poke 'em all.
attached pieces that are easy to detach. That blocky one would be the best one, but I don't know if that's going to be too tricky.
maybe.
Oh, it's moving.
It's moving a little bit. Nice.
Got it, you got it.
That's a good rock. All right, cool.
Bring it up in front
Starboard
of the camera
box.
after the dust clears.
So we've got all of the boxes open,
Get on the lasers.
and
Yeah, that's an excellent sample.
nice. Do a little rotation. Oh, great.
Yeah.
Yeah, that looks really good. If we could also, after we secure the rock, just zoom in from where we collected it and just get a zoom on that,
Yep.
that little ledge. Nice.
So we're thinking starboard box?
Sure, go for
Okay.
it.
All right. Bring up the sample cell though.
Yep.
Coming up.
Is that good with the ledge?
bump out the box.
All right, coming out.
Do you think this will fit in one of the smaller boxes, or do you want to put it in the bigger?
If it'll fit in the little ones, then
Come all the way out.
yeah, might as well
Keep bringing out the box.
All right.
save space.
There we go. Oh yeah, that'll fit.
Okay. Underway.
Nice.
Ching.
That was the most forward one, right?
Yes.
Great.
All right, box coming in.
I'm zoomed back all the way out, Jacob, just for reference.
Yeah, if we're able to just get a little zoom in on the area that we collected and then a zoom out on it, that would be great for some context.
Sure thing. Go back to that one. Okay. It was right here, or up a bit.
Yeah, it's right.
Right
I
above the
think it's,
lasers.
yeah, it was right around there. Yep,
Okay.
great.
Was that easy to break off?
Nice. Could you zoom in a little bit more? Okay, I think that's good.
Okay. Come on. Start coming up this slope.
Coming up.
Yep.
Nice. Yay, we got a rock.
Yay!
Well, and that fit the description of what we're looking for to a T, with in place, perfectly angular, very little crust.
Yep.
Sweet.
Okay. Bridge nav. Can we move 30 meters 160? Thanks.
Large dead sponge stock. Yeah, very sparsely populated this area, and we'll see how this changes as we keep climbing up the flank of this very large ridge feature that is north of the main island group. Starts right around the edge of the sanctuary and extends for about 50 nautical miles. And we had 2 dive sites actually planned on this feature because it's so long and so distinctive. While we're setting up here, just want to welcome our scientists ashore and thank them for their support. So today we've had quite a diverse crowd from different people of the planet joining us. Aaron Judah from the University of Hawaii at Manoa, Sako Matsumoto from the Chiba Institute of Technology in Japan, Ikelau Otto from the Palau International Coral Reef Center, Dr. Christopher Kelly, formerly at the University of Hawaii at Manoa, Dr. James Reimer, University of Ryukyu in Japan, Jonathan Womack, independent historian, and Steve Hauskovich from the National Museum of Natural History, Smithsonian Institution. So thank you so much for joining us and sharing your expertise.
How many viewers do we have joining us from around the world, from the public?
Ooh, let me check. So we have around 52 visitors online.
Nice.
From 7 different countries. Pretty good audience from Palau.
That's wonderful, yeah.
Bridge nav. And we move 30 meters 160. Thanks.
Yeah, well, we're waiting here for our move, and yeah, just maybe wanted to put my colleague here, my Connie Andres, on the spot a little bit, to tell us a little bit about all the research that's going on at the Palau International Coral Reef Center at the moment.
Yeah, thanks for that, Daniel. So at Pickrick, we're doing like multiple projects. The one that we are currently working on right now is the Coral Reef Monitoring Project, where we survey 31 different sites around Palau, ranging from the northernmost state of Kayangala to Angara. And in those sites, we look at the benthic composition, the fish that's living there, coral recruits, edible macroinvertebrates, and also, look at,
This is a cereanthid.
and
So
something
a
else. It's slipping out of my head, but
tube anemone.
so that's one of the projects that we're working on. We also do the MPA monitoring project, which is very similar to the coral reef monitoring project, where we survey all the pan MPAs around Palau. Currently there are 16 that we're surveying, and we survey within the MPA and also on a reference site so that we could see the effectiveness of the MPA. Another project that we're doing is the FAD project, and that's one that Piera is leading, where we deploy baited underwater cameras at the fishing aggregated devices, and then we're seeing what kind of fish are there during certain periods of the month. Yeah, that's just some of the things that we're working on.
Yeah, thanks for that, Mike Honey.
A follow-up question on that, Mike Honey. What is an edible invert?
Very good question.
Shrimps?
So Palauans eat a range of sea cucumbers. So edible macroinvertebrates would be things like clams, sea cucumbers, and sea urchins. Also the trochus. So yeah.
And then has there been any other research done in the PNMS before, aside from eDNA?
Oh, we've also done plankton toes around PNMS to see what kind of larvae are there. But besides that and the fats, we've also tagged tuna in collaboration with TNC. So those are some of the things that have been done within PNMS.
It's a sponge.
Oh, sponge. Dang it.
It was a good guess.
Similar shape.
From there it looked like a long body.
Yeah. This is probably a dictyalis glass sponge.
And another one of our black corals that we collected earlier.
Yeah. Those seem to be pretty, popular around here.
Yeah.
Quite a few of them.
Go for a zoom.
Good representative sample.
Maybe this is a regadrella. So the sponge is in the family Euplectellidae, and there's definitely something inside of it. You see that sort of like little glowy bit?
Yeah.
There's a shrimp in there. The top has a sieve plate, so once the shrimp goes in as a larva, it can't get out. And it lives its entire life in there.
Does the sponge benefit from that?
I don't think the sponge benefits or is harmed by it. It just happens.
Oh my God, it's its own little
like
spongy bubble.
Yeah.
Or bubble.
It's just like an apartment, lots of social distancing.
Although in general, there's quite a few shrimps and crabs or so, they just clean the tissues. They just eat out things off of them, and that's a benefit. Excellent.
There's a little bryozoan too.
And shrimps are cleaners for a lot of different animals, aren't they?
They're very good at their job.
Yeah, very tidy.
I think we got a good shot of this guy.
Yep, all right. Come on.
Bridge nav. We'd like to move 30 meters, 160.
Oh, what's this? What is this?
Thanks. Oh, there's a dead sponge. It's like the same one we were looking at, but this one is no longer alive.
Shrimp is free.
It'd be amazing if something was in there though and just popped out.
That would have been kind of spooky. The way the shadows were happening, it was making it look like there was some sort of like smoke in there. At least my brain was thinking that.
Large polyopogon.
Oh yeah.
And so to all our new listeners and viewers, we are currently exploring an unnamed ridge that is located approximately 70 nautical miles north of Alaska Reef. And ROV Hercules is currently traveling at a depth of 2,137 meters.
Bridge now. 30 more meters, 160, please. Is that an anemone or a cup coral on the rock there?
All bets are in.
Cup coral.
Was that still the Caryophilia?
Oh,
What is that?
Oh, right on the edge.
when to go on mute. Just let it off.
It wasn't as firm as anticipated.
Go for a zoom.
Definitely a cup coral. You can see the hard plates. Could be a Caryophilia.
Yeah, these are hard corals, so they are most closely related to your common reef building corals you would see scuba diving, but this is a solitary coral, so the entire animal consists of a single polyp as opposed to colonial where you have thousands of polyps or connected living on the same colony. That's great, thanks. Yeah, so just looking ahead, so today the plan is to dive until midnight, so we'll have another 9 hours of dive time, hoping to cover some terrain, and then the plan after tonight, after